Professor Marshall was awarded in zoology at the University of St Andrew in Scotland, and then he did his PhD in neurobiology at the University of Sussex, Brinton, working on vision. Then he moved to Queensland, where he works now. He's very interested in study how the animal perceive the environment, especially in marine environment. So the best way is to have to him. Thank you.
Thank you. So I would like to speak without a microphone. So just to check, can everyone hear if I kind of shout a bit? Good. So it's a great pleasure to be here. Thank you very much for asking me. I've never been here before, and it's fantastic so far. It seems like you all do very interesting research, so I'll be interested to learn more throughout the day. Just to introduce myself, that's me, Justin Marshall. I work in a strange place, the Queensland Brain Institute. So I'm actually a neuroscientist but work in the ocean. So my interests are very much to do with how animals perceive their environment. I work on vision and brains and a number of animals. And you can see a couple of those animals here. Here is a mantis shrimp catching a reef fish. So I work on both those animals, and you'll learn, I hope, more about those as we go through the talk. So I'm going to talk about vision in the ocean and a very much a comparative approach, looking at different sorts of animals. So here are some of the animals that I'm lucky enough to work with. This is a mantis shrimp, a Stomata Pod. We also work with sharks, manta rays, mobulids, fruit bats, birds of paradise, turtles, cephalopods, and deep sea fish. So I'm not very good at one thing, I'm quite good at a number of things. It's a sort of, spread your knowledge base. I also run a citizen science project, which is called Coral Watch, in which I try to raise awareness with the general population about what's happening to the reefs. I'm sure I don't need to tell you what's happening to reefs worldwide, because as marine-based scientists you will know, but just to give you the nasty fact up front, we will not have reefs in 40 years' time. They will be gone. The Great Barrier Reef, in the last 2 years, lost 50% of its coral cover. That was on top of a 50% before that. So it's down to 25 or 30% what it used to be. As a neuroscientist working in the ocean, I can't ignore that fact. So I'm now working very hard to help people understand what to do and how to make it better. So as we go through the talk and concentrate on what I hope you agree are the wonderful animals, also think about how we can, as scientists, help to engage the population, because I think this sort of engagement of everyone out there is really important, and we tend to sit in buildings like this and talk amongst ourselves and say how awful it is that everything's disappearing, and then go home and do nothing about it. So my challenge for everyone in this audience is, at the end of today, not particularly inspired by me, but go home and actually do something. Don't go home and think, oh, that was interesting, and yes, I know, and actually do something positive about encouraging people to think about the ocean. So I am a comparative visual ecologist. I work on eyeballs, and here are some of the eyeballs that I've worked on. This is a mantis shrimp. That's a mantis shrimp. That's an octopus, and there's a variety of fish, and even a lizard up there, just to emphasize the comparative nature of what I do. So sometimes I'm called a visual ecologist, because I work on vision and try and work out how the eyes of animals have evolved because of the habitat in which they live. By the way, I'm very informal. I hope that's obvious. So please ask questions. Don't sit there nodding, shaking your head and going, yes, yes, no, no. If I go too fast, if I say things in Australian that you don't understand, please put your hand up and ask me. Or if there's something you're really interested in, don't necessarily wait to the end, ask me a question. If we start talking too much, I'll be rude and tell you to shut up until the end. But, we can at least have a bit of a conversation as we go along. So I'm also very interested in colours. If I also had to say what I do to people, I'll say that I'm interested in colour communication, how animals use colour. And these are some close-up shots of reef fish skin, so the skin of animals that live on the reef. That one's kind of obvious, but this is the tail of a wrasse, this is the chin of a pufferfish, this is the nose of a parrotfish. I know all these things because I took the photographs, but they're not necessarily obvious. But the question for me is, why are animals using these sorts of colours to communicate, and in some contexts camouflage? So, plan of the talk, I said thank you very much, but I'll say it again. It's a really great pleasure to be here, so thanks for asking me. I'll introduce you to the themes of research that we do in the lab, which are basically mantis shrimps, reef fish, cephalopods, deep sea vision, and coral watch. I tend to do this in talks. I say too much, I go too fast, so I'm sorry that I can't go into very much detail, but I prefer to give you a general impression of what we do. So the first theme is going to be mantis shrimp vision. Here is a mantis shrimp leaping out of the sand and missing this fish. As we'll see shortly, this is a very rapid ballistic strike that the animals use to catch prey. Second theme is going to be around reef fish and the colours which they use to communicate or camouflage. And here is a camouflaged fish sitting on the bottom. In fact, this fish is also very well camouflaged, but it doesn't look like it to us. And here is Nemo, the famous anemone fish, in a bleached anemone. So this was in the 2016 coral bleaching. The anemones also bleached, lost their symbiotic algae. This anemone has recovered. So I know this fish intimately. I've taken fin clips from it a few times, put it back, sort of wiggles along and goes, ouch, that hurt, and then carries on. But luckily this anemone has recovered, so there's sort of a good news story there. I'll spend probably most of my time talking about the cephalopods, their brains and vision, partly because this institute, this station has been so instrumental in the past in raising our knowledge about these animals. A little bit on deep sea and deep sea vision, because I spend a bit of my time either in submersibles or on the backs of trawlers pulling up animals from the deep sea. So I've fished as far down as 6,000 metres. This is a mesopelagic net that only goes down to 3,000 metres. I have colleagues that go all the way. So one of my colleagues at the moment is on the deep sea expedition where they're going all the way to the bottom of the Mariana Trench. It takes a long time. Actually, when you get there, it's pretty boring. Don't tell him I said that. So deep sea stuff. And then, yes, at the end I'll fill you with doom and gloom and talk to you about how the reefs are disappearing, but encouraging you to get the population involved in research. Now, what I'm talking about is not just me. There is a great team behind me of PhD students, honours students and postdocs. And here's me looking rather worried because I can only just remember the names of all these people. But I'll introduce more of them as we go along. Fantastic team. The key to success, as I'm sure you all know in life, is to work with people that are far better than you. And all of these people are far better than me. They're really good at what they do, and I kind of go, yeah, that was great. We'll publish a paper together, and that's really the key. Is find someone who's really good and work with them all. So stand next door to them and it rubs off. So I use unusual methods for a neuroscientist. I use ships such as this German vessel. Here is a ship with a submersible on the back that I was lucky enough to have a hand in designing, so I do a bit of ship and submersible design, use submersibles, go underwater as much as possible, live in underwater habitats. So I'm very much immersed in the environment in which I live, which I think is very important. As I said to the PhD students earlier today, we don't observe nature enough these days. We need to spend more time in it. It's all very well to go and get a fish, get an octopus, bring it back to the lab, cut it open and go, yay, I've made a discovery. But what do you really know about that animal? Not much. Spend more time out there. Get the directors of the station, get your grant-giving bodies to give you more money to spend time out there in the world. So, rapidly zipping on with the talk, let's talk about Stomata Pods and their visual system. You've seen this shot before here with the people responsible. So these animals are actually really nasty. They beat up blue-ringed octopus, they strike so fast you can't see it. They come in two varieties, spearing sorts and bashing sorts. They strike so fast that they cavitate water. They break out of aquariums, so if you keep them in glass aquariums, they can easily smash out of the glass aquarium. So very challenging animals to work with. They've hit comic strips because they're so amusing and violent and nasty. So I worked on the harbinger of blood-soaked rainbows, for reasons that we'll find out later. So basically a summary of mantis shrimps or stomata pods. They love light and they love killing stuff. That's all I need to tell you about them. That's it. Here's a fish that doesn't realise it's dead yet. It's been impaled through the brain so fast that it still thinks everything's fine. So they can either impale prey or they beat up prey with these two sort of hardened elbows with which they strike things at really high speed. They fight with each other over holes on the reef and habitat, as well as using these enlarged elbows to crack into other crustaceans and shellfish. They have very unusual eyes. This is a compound eye, a bit like a bee, but you can see it has a special bit in the middle. This is what I spent my doctorate, my PhD, working on, is why does this look different and how is it special? It is the most special visual system in the animal kingdom. They are colourful, so they use colours to communicate, like the fish. And just to go back a bit and remind you why colour vision is important, you don't need it. You can be completely colourblind and still see this apple tree, and you can even see the apples within the apple tree. But colour gives extra contrast and extra saliency, so you can pick the apple more easily. So that's why we have colour vision. This graph expresses our colour vision. Wavelength on the bottom, and I put a rainbow in the background. We have 3 receptors, blue, green and red, with which we can do things like find bananas and fruit. That's us. This is mantis shrimps. They have 12, 4 times as many colour receptors as we have. Why? I have no idea. But I'm making up stuff, and I'm waving my arms, and I'll explain a few things. But we have no positive proof as to why these animals are so unusual. But all of these colour receptors are contained within that special mid-band region of the eye. So that's the specialised part that contains 12 colour photoreceptors. This is a confusing graph, I'm sorry, but it just says one thing. On the y-axis here is wavelength discrimination. So you can train animals to tell you how good they are at discriminating colours. Now I can discriminate your blue shirt from that blue seat because they're slightly different shades of blue. I'm good enough in the blue to discriminate that. You can ask animals like goldfish, honeybees, humans and butterflies to tell you how good they are at discriminating different colours across the spectrum, from blue to red. And this curve here, the green dotted curve, is a theoretical curve of what stomata pods should be able to do. Because they've got all these colour photoreceptors, of course, they are the best at discriminating colour in the world. No, they are the worst. So this is their discrimination curve. It's far worse than any other animal that we've tested. It's not this, it's this. So their colour vision is actually being conducted in a very different way to ours. In order for me to see colour, I have to compare 2 photoreceptors. So my blue photoreceptor, my blue cone, is very excited when I look at this blue chip. My red photoreceptor is not, and the ratio, the difference between those 2 excitations tells my brain I'm seeing blue. Mantis shrimps actually construct colour in a frequency analysis system that's more like the way in which the ear works. I'm going to leave it at that and move on to the next subject, but you can ask me about that later. The next subject is that these animals, including their relatives, the crabs, stomata pods, see polarized light. And they see polarized light in more detail than any other animal. Again. So in fact, one of the things that we've done recently is build this camera. It is a bio-inspired camera. The CCD array of which is inspired by mantis shrimps. And what the camera does is allow us, polarization blind humans, to see where there is polarized light in the environment. So this picture of a mantis shrimp in its hole tells me that the red here is the red appendages, the maxillipates, on this mantis shrimp are reflecting polarized light, as well as blue light. So this animal is talking, it's getting signals in the blue and in polarization with these maxillipates, and they actually show these off to each other when they meet. And they're saying something very specific to each other about who they are, whether they like McDonald's or not, whether they go to the movies, whatever it is mantis shrimps talk about. They speak with polarized light, as do other animals. The way in which they speak, or the way in which they analyze ordinary polarization, is with these parts of the eye. So remember, this is colour, and these parts of the eye conduct polarization vision. Just to remind you, light travels in waves, and light from the sun travels in waves in all possible directions. Light becomes polarized when it's reflected or filtered. Some of you have polarizing sunglasses on that filter polarized light. Here is a vertical polarized filter beam, here is a horizontal. So the light reflected off the sea surface or the road surface is horizontal. We use polarizing sunglasses to remove glare. These animals analyze linear polarized light in more detail than any others. Very, very, very confusingly, they also analyze circular polarized light. And I don't have time to go into the physics of that, but if you want to talk to me afterwards, I can bore you with lots of quarter wave retarded talk and all sorts of physics and stuff. But circular polarized light is light that travels instantaneously in one direction, but it also rotates as it comes towards you, clockwise or anti-clockwise. We didn't know that animals did this, but as physicists and humans, we've been using circular polarized light for a long time. And the animals, the mantis shrimps, these are the photoreceptors in longitudinal section, have a special photoreceptor that turns circular light into linear so they can see it. And it turns out, why do they do this? It's all to do with sex. It's all to do with mating displays, because the males reflect circular polarization on their tails, which the ladies like for some reason. Again, we're not quite sure why the ladies like this, but as humans, we're often confused about, as a human male, I'm often confused about why ladies like certain things and don't like other things, and that's how you learn about life. And mantis shrimps learn about the ladies and the gentlemen by looking at differential reflections in polarization. So the take home here is that animals, such as mantis shrimps, but also other animals, use polarization to talk to each other. Now, luckily for me, we can then use their vision for areas of application. So, via inspiration of camera design, their circular polarization vision also enables us to encode data on CDs and DVDs better. Again, I'm just going to go through this really quickly and make a note and talk to me about it afterwards if you're interested. We can use mantis shrimp inspired cameras to detect cancer. So early detection of cancer, either on skin or at the moment my colleagues in the States are using a mouse model and basically sticking a camera up the mouse's bottom in order to look for colon cancer, because that's of great concern in countries such as America where they eat too much meat. And if you eat too much meat, then often you get colon cancer. Mantis shrimps can tell us where the cancerous cells are, because cancerous cells reflect polarized light differently to normal cells. Another area of application is a project called Prawns or Shrimps in Space, where I've convinced satellite engineers over a number of years that these animals have a better way of seeing the planet than do satellites, because in fact their eyes scan over the world, they move over the world in exactly the same way as a satellite moves over the Earth. The way in which they take in information is just like a satellite. And satellites are used to look at coastal areas such as this, or reef areas such as this, Heron Island. So we use the mantis shrimp vision to tell us about ways in which we can design satellites better. The fourth application area, which we've only just published, is that when the satellites go down, when America turns the satellites off, and you can't use a GPS anymore, all you need is a mantis shrimp in your pocket. It will tell you exactly where you are on the surface of the Earth, if you ask it nicely. Again, I'm not going to go into details, but it's basically using the way in which they see polarized light on the water to geolocate on the surface of the planet. So So all of these application areas are nice because it enables us to get money to do research on them. I don't really care because I like the biology. And what we're doing now is trying to look at the nerves beneath the retina and work out how the retina here, you can see in the eye, sends information down to the centralized brain. So here's the retina, the eyes. Here's the centralized brain. We're working very hard on the neuroanatomy and the physiology of their brains. So moving swiftly on to the second. Yeah.
Do they have predators in nature, and do their predators have the polarized vision?
Good question. They do. Their predators are very often the cephalopods, and they have polarization vision. And I'll say something about that shortly. So hold that question, but yes, they do. And there's probably an arms race that has been going on between Stomata Pods and Cephalopods for 400 million years. So they've tuned their polarization vision, and that may be one reason why Stomata Pods have circular polarization reflection, so the cephalopods can't see that. So it's a good way of private communication in a waveband that your predators can't see. But yes, we'll get into that a little bit more after I've whipped through the reef fish. So reef fish vision. We ask questions about colours for communication and colours for camouflage. Here's the happy team of people working on reef fish vision. We measure the colours of fish, so these curves are now not sensitivity, but the reflections of these various fish. We're interested in colours for communication, colours for camouflage, so here's a variety of camouflage strategies of fish. One of the things that we realise in doing this is that fish see the world very differently to us. These are the spectral sensitivities of this fish, a little damselfish. It sees in the ultraviolet, part of the spectrum that we don't see at all. Down here, here's the rainbow again for us. Here's our sensitivities from blue through to red. These guys see in the UV. Why? Because they talk to each other in ultraviolet. So if you look at this picture of damselfish in human black and white, if you like, and here are the human spectral sensitivities. Now look at that fish in damselfish, where the added contrast here is because of the ultraviolet sensitivity of this fish. So this photograph is taken with a UV sensitive camera that sees the contrast in this fish that we cannot see. So again, these animals are using a means of communication that big animals such as ourselves, and in fact big predators such as the big fish in the ocean, also can't see these contrasts. So they're using a private communication channel to talk to each other that is invisible to animals such as ourselves, or barracuda, or other big predatory things. One of the things we're doing with reef fish at the moment is training them to do a colour vision test. Have you all done this at the eye doctor? It's the Ishihara colour vision test. They ask you to spot a number or a word, and we ask the fish to spot a simpler task, which is a single coloured dot in the midst of others, under which we place food. They can peck through the paper, get the food. So that tells us what sort of colour vision the animal has, and we can plot that in an animal colour space. We also get the animals to show us that they suffer from illusions. So this is an illusion. What colour is that square?
Brown.
Brown. What colour is that square? Orangey yellow. In fact, they're both the same colour. They're both brown. If you put a mask over, you can see that this is an illusion. You appear to be orange because it's in the shade. And our colour vision system makes our brain think it's orange, whereas in fact it's brown. So what we do with the fish is ask them to do the same slightly modified test. We have trained this fish to peck at an orange spot and get food. It looks at this and goes, whoa, is there an orange spot there? No, this and this are the same colour. We ask it, does it suffer from the same illusion? And yes, it does. So it pecks the orange spot and then gets a food reward. And that's very interesting from a psychophysical point of view, because psychophysicists will tell you that illusions are a top-down phenomenon where the big cerebral cortex in our brain is setting up the illusion. Fish don't have a big cerebral cortex, so the illusion is probably set up within the retina. Maybe it is in us too. My cognitive colleagues are very challenged by this. So it's an interesting way of dissecting colour vision in fish. The other thing we do is dissect colour vision in fish with molecular mechanisms and mapping the retina. I'm going through this very quickly, I'm sorry, but we look at the opsins, the visual pigments of a variety of reef fish, where they lie within the retinas of reef fish. We've also recently published this in Science, which is looking at the retinas of deep sea fish, because, of course, deep sea fish only have a single visual pigment with which to see blue, because they live in a very blue world. They have blue bioluminescence to look at. They need one visual pigment, except for this bloody fish that has 40. So this fish has 40 visual pigments, or it's close. This one doesn't actually. It's directness which has 40, but there are 3 different deep sea fish that have multiple visual pigments, which is really weird because they don't need them. And don't ask me why, because you don't know. I can make up a bunch of ideas, but we really don't know. So there's a mystery. But as soon as you start looking at the molecular side of vision, you come up with a whole bunch of really interesting things that you have to then work through. And we do that with reef fish, deep sea fish, anything we can get our hands on. So what about the cephalopods and this third theme in the lab? We spend a lot of time working on octopus, squid and cuttlefish. In particular with this gentleman, Wen-Sung Chung. He's an astonishingly good Taiwanese scientist. During his PhD, he wrote an entire guidebook with his previous mentor on the cephalopods of Taiwan. A 500-page guidebook in Mandarin and English, and did his PhD too. Who would do that? So that's a challenge for all you PhD students. You've all got to write a book each on whatever it is that you're working on. He's very good. He does a lot of this work that I'm going to describe. He's got an amazing knowledge set in terms of the cephalopods and their natural history. He's works on aquaculture, all sorts of things. And I also have 2 PhD students, which I haven't put their names there, but Italy and Mexico. So this is an Italian PhD student, Gabriella, who works with me. This is a Mexican PhD student, Alejandra. And essentially, Wen-Sung at the moment is looking at squid brain, octopus brain, and cuttlefish brain. So that's how we divide up the work on these amazing animals. And you may recognize this painting because it's from here. Just to show you this video from Roger Hanlon, which I'm sure you've all seen. Right at the back. Where's the who hasn't seen this? Everyone's seen it, yeah. But it's pretty amazing. Now, where is the octopus? Where's the octopus? Woo! There it is. Yeah. But the amazing thing is they do that camouflage while being completely colourblind. Here is the single visual pigment of a cuttlefish, same for octopus and a squid. They have no access to colour at all. They cannot use colour to camouflage themselves. So this animal is camouflaging itself while being completely colourblind. Here is the same animal 30 seconds later displayed. You can get an idea it's colourblind by the fact that its displayed colours are in black and white. It's not using bright colours to communicate with. But how do they do this? It's basically through evolution. They can match the background of likely ocean bottom. It's basically greens and browns and those chlorophyll-like things. Because if you play an unfair trick on them and give them a gravel bottom in the bottom of an aquarium like this, they don't go blue and yellow because they can't see the colours. But more importantly, they can't see the contrast here. These are very obvious contrasts to us. Like the apple in the tree, the red apple in the green tree, obvious contrast. A blue piece of gravel and a yellow piece of gravel, obvious contrast. But this animal thinks it's on an even shade. So if you look at this photograph, but in black and white, there is no contrast. It is putting on a body coloration pattern, which it believes camouflages itself against the background. So this is actually some work I did with John Messenger, who's visited this station many times, a while ago in Plymouth, and we used different coloured gravel to say again and again, and people have to say this over and over again, these animals do not have colour vision. They are completely colourblind. And that includes octopus, squid and cuttlefish. So, just to give you a little history lesson, I'm sure you all know this, we've now begun to delve into the brains of the cephalopods in great detail, and going back in time, Ramon y Cajal was the first to do exquisite silver stains of the cephalopod brains. J.Z. Young, who worked here also, described octopus, squid and cuttlefish brains. Then moving through the years, you've got people like Boycott, Hodgkin, Huxley used it as a giant axon model. Then guys from here, Benny Hoffner. And what we're now trying to do is contribute to this using MRI, high-resolution MRI, and those are the next few slides that I'll show you, to look at the brains of octopus, squid and cuttlefish, and work out what the wiring is behind the brain to make it a so-called connectome, or at least a predictive connectome of what connects to what in the brain. So we put squid, octopus, cuttlefish into an MRI, as you see here, but actually a much more powerful magnetic resonance imaging machine than we would use for a human. So it basically fits a small cephalopod brain, and we can then begin to work out which parts of the brain connect. Again, I'm not going to go through the details of how that works, but I can talk to you about it afterwards if you're interested. But we can begin to build up a connectome of what's going on and look at the parcellation of different optic neuropils within the brain. Here's a squid, here are the eyes, here's the brain being imaged. So we can look at these different parts of the brain, label them according to different factors. But then, if you like, more importantly, we can begin to build up connection maps. So here is the optic lobe, the biggest optic neuropil, and its connections through into the main brain of Cephalopods, which is a squid. Then here are some other connectome informations from, again, the optic lobes feeding into the central brain. And this is all from MRI imagery at very high resolution, 16.4 Tesla resolution, which gives us confidence that these are actual nerve tracts. We also follow it up with silver stains and fluorescent staining to make sure that this predictive imagery is truth. The result of that is that we have a tractography map where we plot all of the different neuropils and their connections. So where you see a red square, it means there's definitely a connection between here and here. Compare that to the 282 tracks that J.Z. Young mapped just using silver stains, an amazing body of work, and we found an extra 153 interlobe tracks. So we also have confirmed all of these that J.Z. Young found, so we believe that the methods we're using are a good new method of mapping the brains of these amazing animals. So we have, of course, not just, it's no good just to map the connections, but you then have to go, all right, here are the connections, what could they possibly do? So we're looking at functional aspects of that, including visual motor control systems, and really you won't understand this, but it's just basically showing that there's lots of connections between different parts of the brain that we didn't know about earlier. We've discovered a second chiasm which wasn't known about, which implies that the animal has exquisite knowledge of both its legs and its camouflage patterns, which was actually predicted against previously. So the animals are able to see themselves and know where their arms and legs are based on this new MRI information. As well as comparing octopus, squid, cuttlefish, we can also compare different types within one group. So this is a midwater octopus, a solid